Hi all, Dr. Clark here again. For natural resources, we're going to talk today about chemistry of biomolecules, or the chemistry of molecules that are created by life. Now, some of the molecules that we're going to talk about them being directly harvested as a natural resource. For example, we'll talk about protein, and maybe we're going to talk about harvesting living creatures for that. Chicken, beef, sheep, whatever it might be, as a natural resource, and getting the protein from those. Same things with lipids, things like that. Okay. The other things that we might be talking about are like complex carbohydrates like cellulose, and getting that as products or natural resources from forest material, from trees. Okay, so there's the collection of these natural resources off the environment, and then we're going to talk about situations where we know enough about the chemistry of the biomolecule or the pathway of the biomolecule that we can manipulate it and create our own synthetic biomolecules or synthetic chemistry for that purpose. And we'll talk about situations where that's been done, or situations where we know the process and we've created a similar process, like solar panels, and the fact that solar energy is basically the same premise as photosynthesis. Okay, and we'll talk about these kind of things as we progress. But first, today, we're really going to talk about biomolecules and the chemistry of these molecules. Okay, so first of all, biomolecules are made of macromolecules, or they're typically called macromolecules because they're large. Okay, and there are 4 macromolecules or 4 biomolecules that we're going to talk about. Now, there might be some deviations from this depending on who's lecturing or who provides the information. But these are the 4 main macromolecules, biomolecules that are building up the body of an organism. Okay? And then we can utilize these in different ways. Okay? And those are proteins, nucleic acids, carbohydrates, and lipids. Okay? Now, macromolecules are composed of monomers, or smaller components. Okay? So each one of these macromolecules has smaller components that it is comprised of. Okay? And we'll go through what these components are as we progress. Okay? But when you link these monomers, these smaller subunits, into larger subunits, they're typically called a polymer. So sometimes people refer to macromolecules as being polymers. Okay? And we'll talk about this later as we progress and talk about polymer research, okay, or macromolecule research. So there are 5 general functional groups. Okay? So carbohydrates will end in a hydroxyl, lipids will end in a carbonyl, proteins will either end in an amino or have an amino group, okay, that's in the middle, okay, or they'll end in a carboxyl, or sometimes both. Okay? And DNA will have a phosphate off of typically off of some sugar. Okay, and ATP is the same way. Okay, and so each one of these has their own group, and each one of these has their own general properties. And we'll get to this when we break off each one of the macromolecules. So the first one that I want to talk about are proteins. Okay? Now, proteins, as most people know, are hugely important for our diet, but what we often don't realize is proteins are what makes you you. Okay? So your hair color, your eye color, skin tone, shape of your nose, shape of your lips, your fingers, your height, all that is really just the compilement of proteins. And these proteins are made of subunits called amino acids. These amino acids are linked together with what we call a peptide bond, okay, which is just a bond between nitrogen and carbon. Okay, it's kind of a specialized bond that allows for amino acids to be bound together. Okay, the more amino acids you get together, the longer the chain. okay, and you get yourself a simple protein. We'll come back to the shape and structures of proteins in a little bit. Okay. As you increase the size and bind more amino acids together, your monomers, okay, are building into a polymer, okay, but then that polymer has a special name, which we call a polypeptide, okay, meaning multiple peptide bonds or multiple peptides. So another word for proteins is polypeptides. So here you can see how that peptide bond is formed. You can see we have an amino acid over here, okay, with a hydrogen bound to a nitrogen, okay, an amino acid over here with a hydroxide bound to a carbon, and this comes together. Energy is supplied to make this bond work, but water is removed. It's called a dehydration reaction, okay? And you get a carbon-nitrogen bond or a peptide bond, okay? And this forms a polypeptide chain. Now this can happen over and over again, because here you can see off here you have another OH, here you have another hydrogen, so you can get another dehydration reaction off this side and this side, and it can just continuously build and build into a really long chain of amino acids. Okay. So amino acids, or that small subunit of a polypeptide, are interesting for a couple reasons. First, they consist of three main things: an amino group, a carboxyl group, and then a functional group. Now, all amino acids will basically have the same amino group, the same carboxyl group, and then their functional group will be different. And the functional group could be as simplistic as just a hydrogen, or as complex as, a whole strain of, more carbons, or another carbon ring or something like that. Okay, so the interesting piece, though, is that functional group is really what gives that amino acid its unique properties. Okay? And there are 20 different amino acids that occur naturally. Okay? I believe chemistry has created a few amino acids. I think they've created 4, but maybe 2. I'm not really up per se on amino acid synthesis, but I know chemistry has created a few amino acids on their own. Now, we're not really going to talk about synthetic amino acids right now. Okay? The 20 amino acids, this is kind of another one of those interesting pieces about proteins. Those 20 amino acids are the same 20 amino acids in every organism. So if we're talking about bacteria, okay, they're built of the same 20 amino acids as humans. We're talking about plants, same 20 amino acids. We're talking about fungus, same 20 amino acids. The other interesting piece is, if we see a meteorite that crashes into the planet, and we go and swab that meteorite or that asteroid or whatever you want to call them, that foreign body, okay, we found numerous amino acids on it, and they're all the same amino acids. Okay, I'm not entirely sure if we found every single amino acid that we know on them, but we found lots of different amino acids on these. So it suggests that these amino acids may have, came here on asteroids or meteorites, whatever, okay, and smacked into the planet. That's how they might have got here to begin with, maybe, okay. But we don't really know what it means, but we know that it means that the amino acids occur off this planet. Okay? So the building blocks for proteins occur elsewhere. Okay? And that can be a fairly interesting piece, and we can talk about it more maybe in a evolution of the planet talk or something like that, or another class that you might take. Okay? Now there are 4 general levels of protein structure: the primary level, secondary level, tertiary level, and quaternary level. Now the primary level we already talked about. It's just amino acids linked together with peptide bonds between them. So it's a polypeptide chain. It's just long amino acids. Okay? Secondary structure is when those amino acids twist or kink together and form hydrogen bonds with themselves. So the secondary structure is often distinguished by things like a helical shape or a sheet shape or a coil or something like that. A tertiary structure is when those helixes and those beta sheets, when they start to come together and they form a complex protein. Okay? And tertiary are 3D shapes. Okay? And quaternary is when you take a few tertiaries and you link them together. Okay? So you can see as we progress, primary, secondary, tertiary, quaternary, you get more and more complex. Okay? And so here you can see what I'm talking about. So here, primary structure, amino acid chain. Okay? And then you can have the alpha helix or a beta sheet, okay, hydrogen bonds between them. Okay? But then if you have these alpha helixes and beta sheets, they can come together in a tertiary structure. Okay? Then you might take these tertiary structures and get a few tertiaries bound together to get a quaternary structure. Now, this is all good and this is all important, and the reason why it's important is when we start looking at the link between nucleic acids, or DNA, and proteins. So DNA codes for your proteins. Okay? So the DNA sequence that you have in your genome, all 25,000 genes, roughly 25,000 genes, okay, is going to code for an amino acid sequence, each one. Okay? Now, you would think, well, if that's the case, we only have 25,000 proteins. But in actuality, we have discovered at least 150,000 different proteins. So that means that the primary structure is based purely on your genetic sequence. But these other structures might not be. Okay? Whether it forms an alpha helix or a beta sheet, whether the tertiary looks like this or in a different form, what's the quaternary structure look like, this might be determined by what's available in the system. Okay? Or it could be determined by environmental effects. Okay? We're not entirely sure. Okay? Some things we know, but other times we don't really know why we have so many different proteins and so few genetic codes. Okay? One man that might have known, or at least worked on this, is a man named Linus Pauling. Okay? Now, I highly suggest, if you're interested in scientists and the history of science, that you pick up Force of Nature. Linus Pauling won 2 Nobel Prizes. Okay? He's the only person to win 2 Nobel Prizes by himself. Okay? And so there are only 4 people that have won multiple Nobel Prizes. Okay? Barton, Curie, Pauling, and Sanger. Okay? Now, Pauling is the only one to win them by himself. Okay? He won one for discovering the secondary structure of proteins, or the helical shape in the beta sheets, so structures of proteins. He won one Nobel Prize for that, and another Nobel Prize for peace activists during times of war. Okay? Now, Linus Pauling has a really interesting story about not finishing high school and dropping out of high school, getting a degree when he was really young, going to the University of Oregon State University, and all this. I highly suggest, if you're really interested in a history of scientists, that you read Force of Nature. Excellent book, and Linus Pauling's story is really unique. Okay? The other piece of this is the link between protein shape and nucleic acids. So we're going to start talking about nucleic acids, and then there's this book that was written by James Watson, who was one of the biologists that discovered the structure of DNA. Okay? And I also highly recommend this book, okay, on the genetic revolution, or the structure of DNA. And it's these 2 books that really, and these 2 individuals, along with a lot of other people that we will talk about later, okay, when it comes to DNA, but it's really these 2 that were kind of in the race to figure out what shape was DNA. Okay? Linus Pauling was wrong. He thought it was a triple helix. James Watson was right with others. Okay? Francis Crick, Rosalind Franklin, Maurice Wilkins, with information from Erwin Chargaff and all these other individuals, really helped to figure out the structure of DNA. Both excellent reads. I highly suggest them. Pick them up, read them. I don't want to say they'll change your life, but if you're into science, they'll change your life. Okay? All right, so nucleic acids. Nucleic acids are really simple structures in the sense that they're built of nucleotides. Okay? And nucleotides consist of 3 parts: a 5-carbon sugar, a phosphate group, and then some organic containing base, okay? Which we call nitrogenous bases, because the nitrogen piece there. Okay. Now, depending on what type of nucleic acid we're talking about, will depend on what nitrogenous base or nitrogen containing base we're talking about, and what sugar we're talking about. The phosphate group is identical in both types of nucleic acids. There are 5 different nucleotides, okay, and really they're mainly determined by what is the nitrogenous base and what is the sugar type. Okay? And we'll look at these in just a second. Okay, so the 2 types of nucleic acids, you're probably familiar with these because you've heard about them many times, is deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. Now, the 2 structures are very similar, except for RNA has uracil, the nitrogenous base uracil, instead of thymine, which you find in DNA. RNA is also often just in a single strand. Now, it can be found double-stranded, so it can wrap on itself and form what we call a hairpin loop. Okay? And in some cases, like transfer RNA, it can be in a double helix form, at least portions of that molecule in a double helix form. But RNA itself mainly is just one-stranded. Okay? And then the sugar for RNA is ribose and not deoxyribose, okay? Which means off the second carbon, you have an OH group for RNA, which makes it a ribose sugar. And if the second carbon, you have a hydrogen, okay, that makes it deoxyribose sugar. Okay? So what is interesting about DNA is the structure of DNA, as most of you know, is double helix. Talked about James Watson, Francis Crick, Rosalind Franklin, Maurice Wilkins, a bunch of other people that had a huge part in playing a role in figuring out that it's double helix. Okay? Well, in part, it's double helix because of the pairing of the nitrogenous bases. Adenine always pairs with thymine, and cytosine always pairs with guanine. Now, this is called Chargaff's rule, okay? Because Erwin Chargaff figured this out a long time ago, before we knew that DNA was a double helix. He figured out this pairing mechanism. But the nice thing about that is adenine, okay, has 2 carbon rings and thymine has a single carbon ring, okay? Which means that you have 3 carbon rings at any point where adenine and thymine come together. Guanine also has 2 carbon rings, and cytosine has a single carbon ring. So you have 3 carbon rings where they come together. So the DNA, the double helix, is uniform in width. As you progress or add nitrogenous bases, nucleotides together, the width is identical or nearly identical. Okay? Which makes it useful, and you'll see why later when we start talking about GNOs and genetically modified organisms and the natural resources behind those. Okay? These guys are held together by hydrogen bonds. We already talked about hydrogen bonds, these kind of weak forces between a hydrogen and the negative or the polar side, the negative side of another atom. Okay? And adenine and thymine have 2 hydrogen bonds between them, and cytosine and guanine have 3 hydrogen bonds between them. And then these nitrogenous bases being linked to a sugar. Okay? Then the sugar is also linked to a phosphate group. Okay? which creates what we call the sugar phosphate backbone. Okay? And the bond between the 5th carbon in the sugar ring and the phosphate is called a phosphodiester bond, or what sometimes is called an ester bond. Okay? Not hugely important, but might be important when we start talking about genetically modified organisms, because we need to know a little bit more about how DNA is copied and how we can modify DNA. Because remember, natural resources are about things that organisms make. Well, nucleic acid is one of those things. So DNA is one of these things that organisms make. And as we progress into the future, I assure you that we'll be utilizing more DNA-driven products, okay, than anything else. Because we can modify the DNA. We can get organisms to make things for us through changing their DNA. And we'll talk about this as we progress. Okay. Here you can see the double helix, okay, uniform in width all the way. There you can see that double bond between adenine and thymine, triple bond between guanine and cytosine, and this phosphodiester bond between the phosphate group and the sugar. Okay. All right, carbohydrates. Okay. Carbohydrates are monomers that really play a critical role in energy. When we start talking about energy and we start talking about energy of living organisms, we have to talk about carbohydrates. Carbohydrates are the most efficient energy storage that we kind of have, or at least the most efficient conversion of energy. Okay? And carbohydrates really come in a very simplistic form, typically, okay? And that is this ratio of 1 to 2 to 1. So you have 1 carbon for every 2 hydrogen for every 1 oxygen. Okay? And this is just the typical ratio of any carbohydrate. Okay? Now, the interesting thing about carbohydrates is, you know, they come in many different forms, just like you have proteins in their primary, secondary, tertiary, quaternary. Carbohydrates sizes vary also, and they vary mainly by, are they simple carbohydrates, and so they might consist of 1 or 2 monomers, or are they complex carbohydrates where there might be a long chain of multiple monomers. We'll look at it in a second. Simple carbohydrates are typically small, and small carbohydrates means fairly easy to break down. Okay? One of the simplest carbohydrates, and one that we often talk about, is glucose, C6H12O6. Okay? It's a monosaccharide, okay? And it is very useful from an energy perspective. Okay? Because the nice thing about carbohydrates is you have this carbon backbone, okay? And then you have this large amount of hydrogen bonds, okay? Which means that every time you have hydrogens bound to carbon or oxygen, you have bond energy. Okay? And so the more hydrogens you have in a solution or in a molecule, the more energy you have, typically. Okay? And so that's what makes carbohydrates this really rich, energetically speaking, rich molecule. But it's also easy to break down. Our body has evolved, and other organisms' bodies have evolved to be able to digest things like glucose. Okay? Now you can also have disaccharides, okay, which are still simple carbohydrates and just consist of maybe a couple monosaccharides. So you get things like sucrose. This is table sugar. Okay? And that's just really the combination of glucose and fructose. Okay? These other simple monosaccharides bound together to make this a little bit more complex sugar. Now, as you bind things together, complexity increases. Therefore, so does the energy. But so does the amount of energy to create that molecule. Okay? So remember, to make bonds, you have to supply energy. Okay? And so if we're going to bind glucose and fructose together, we have to add energy to get sucrose. Okay? Now, the other thing, though, by doing that is often those little bit more complex molecules are better, have better storage purpose. So they store for longer. They don't break down as fast naturally as things like glucose and fructose. Okay, so that's one real caveat, a real nice piece of carbohydrates, is they have the ability to chain together to be much more complex, and that complexity allows them to be stored for a longer period of time. So when we talk about stored energy, plants and animals store energy a little bit different, okay, than each other, but it's really the same kind of premise. You take glucose molecules and you're just linking them together, or fructose, or some of the other monosaccharides. You're linking them together. Plants store carbohydrates in the form of starch. Animals store carbohydrates or polysaccharide chains, so complex carbohydrates, in the form of glycogen. But even further than that, okay, you can build on those carbohydrates and make them even more structurally sound, okay, by making big polysaccharides with even more bonds and sometimes more specialized bonds. Okay, and you can get things like cellulose. Okay, is what really, if you look at vegetation, if you look at a tree, okay, and you look at wood is really just cellulose. It's just those wood fibers are really just complex carbohydrates. Okay? Chitin, which is the exoskeleton or the material for exoskeleton of invertebrates, so insects and mollusks and crayfish and lobster and a lot of these other things, their exoskeletons are made out of chitin, which really give them this structural system, okay? And it's no longer really used for energy storage. Nonetheless, still very important, okay? But not used for energy storage. Okay? Now lipids, okay? Lipids, otherwise known as fats or other molecules, okay? The interesting thing about lipids is lipids are not soluble in water. Most people know this. Okay? They're not soluble in water because no hydrogen bonds can form. Okay? Because lipids are nonpolar. So they don't have a positive or negative side to the molecule. Okay? So water can't bind to it. Okay? And so you can't form a hydrogen bond, and therefore they cannot dissolve or be saturated in water. And so there are lots of different types of lipids, and this is where a lot of people just think, well, lipids just means fat, okay, or maybe oil, okay, but there's lots of things. Steroids are types of lipids, rubbers, wax, pigments. There's all kinds of things that consist of lipids, or nonpolar molecules that are not dissolvable in water. Okay? Now the properties are really important because the fact that they're not dissolvable in water allows for them to be good storage molecules. So if you consume too much glucose or too much carbohydrates, you can then convert that carbohydrate into a long-term energy storage, okay? One that's not likely to dissolve. Now, it's hard to break down, as many people in the United States know, since we're, like 65 to 70% of the people in the United States are overweight, I think is the last thing that I read. Okay, but nonetheless, so we know that it can be very difficult to break down fats after they are created. But the evolutionary point of that is excellent, because in most circumstances, energy is limited. Okay, so if you're, say, a squirrel or something like that, and you have to eat a lot of energy today, okay, before winter, then you would consume a massive amount of energy, more than you could ever burn, and you'd store it as fat. Okay? And that's long-term energy storage. It's not dissolvable in water, so it's not going to easily break down just over time. Okay? You're going to have to be able to convert that fat back to energy through enzymatic processes by breaking it down. Okay? And that's what makes it very unique and a great energy storage system. Okay? So fats really have two subunits: a fatty acid, or what we often call fatty acid tails, and glycerols, which sometimes are called glycerol heads or glycerol backbones. Okay? The fatty acid tails are just chains of carbon and hydrogen atoms, which are known as hydrocarbons. Okay? And again, just like carbohydrates, the more carbon-hydrogen bonds you have, the more energy you have, because any bond represents energy, represents potential or bond energy. Okay? And then at the end of the carboxyl is typically, or at the end of the chain, the fatty acid chain, is a carboxyl group. So here you can see a really common fat, and a really common diagram of fat. Okay, most fats that we talk about, like you look at the back of a package and it says there's a certain amount of fats, or you get your fat content of your body and it's a certain percent. Really, it's talking about triglycerides. Okay, and it doesn't mean that fats don't occur in other forms. There's monoglycerides and diglycerides. Okay. But most of the fat molecules that we talk about are in the form of triglycerides, which means they have these 3 glycerol pieces, okay, and can be called heads. Sometimes it's called backbones, and then off of them you have 3 fatty acid tails. Okay. Now, whether they are considered saturated or unsaturated really comes in the form of the tail, so that the head or the backbone, the glycerol, doesn't really change. Okay, it's these tails. So if you have the most, or a saturated fat is a molecule that has the most hydrogen bonds possible. So all the possible hydrogen bonds, or all the possible hydrogens bound to the carbons, exist. Okay, so all the bonds are filled. Okay. A unsaturated fat is when some of those electrons are shared between carbons. So here you can see double bond. So saturated fats are solid at room temperature, and unsaturated fats are liquid at room temperature. Okay, and it really has to do with the property of, are there double bonds between carbons? Okay, so do the saturated or fatty acid tails, do these tails have kinks, or are they free with no kinks? Okay. And that's really where saturated versus unsaturated comes in, and it depends on the report that you read and who you talk to from a nutrition standpoint on which one's better for you, which one's worse for you. But that's where saturated versus unsaturated comes in. Saturated means it's saturated with bonds, single bonds, and unsaturated means that there's double bonds between carbon atoms. Okay? So those are the 4 biomolecules that we're going to talk about. We'll talk about situations where carbohydrates are very important for things other than diet. So maybe they're important for cellulose production, wood products, things like that. We'll talk about situations where DNA or nucleic acids are important for designing organisms that can do things like maybe break down oil products or break down lipids. So maybe we use things like bacteria or algae or something like that, and we change their genetics to be able to break down oil spills. Okay? And we'll talk about situations where really these bioproducts are just a result of we need more food. So we need to either increase the amount of protein on the landscape. So do we do that with chickens? Do we do that with beef? Do we do that with fish? Are we doing that with insects? Okay. Maybe that's the next step. But anyhow, that's what I'm going to talk about in the future when we talk about biomolecules. Okay. So see you next time.